Satyajit is a well-known Indian immunologist, great teacher. He has been part of these series of infectious disease meetings in the past. He was here last in 2010. And he will teach basically by some vigorous hand-waving as well as board work. And he will tell us about topics he's interested in immunology. Thanks.
Okay, I was going to say to Gautam that he shouldn't really make people waste energy by moving, since there is a little saying about prophets and mountains, and mountains go to prophets, prophets go to mountains. So I thought there's really no need for me to go up there, so I can sort of walk up and down here, depending on how dispersed people are sitting. This is not, as Gautam said, my first time in talking to this group, and yet I never quite get over my sense of uncertainty about what to talk about, and I keep trying different things each time. This time, since Vinita's, I gather, done a certain amount of the nuts and bolts of immunobiology yesterday, I was thinking today morning that what I would like to do is to talk about problems in our understanding of infection and immunity that I think could use a modeling approach, and yet to my knowledge such approaches have not been enthusiastically, or at least have not been iteratively used, meaning that there is a model, that there's a prediction from the models that's been tested by wet work, the model's been refined, and so on and so forth. There have been some a priori models published over the years, but not with any real connectivity. So let me start with a hypothetical scenario that I've used when I'm teaching immunology to biology students. And that is, the immune system you expect to be able to deal with incursions of microbial pathogens from the outside. When you think about the body design of vertebrate animals, or invertebrate animals for that matter, you think about it in terms of location of the physiological system, the heart, the liver, the gut, in a particular three-dimensional space position within the body. That's what biologists mean by body design. Biologists like to pretend that body design is optimal, which it clearly is not. All that they mean is that body design is subject, at least in some components of it, to some discernible evolutionary selection pressures. However, if you think about the immune system, if what you were trying to design bodies for, or if you're trying to design the location of the immune system in the body for, is dealing with microbes coming in from outside, where would you locate the immune system? If you are doing, you are sort of playing the blind watchmaker. Everybody's heard of the blind watchmaker, I presume. No? Yes? This has to be conversational. We ain't going nowhere without that. So everybody's heard of the blind watchmaker, right? The Dawkins argument about how despite a lack of purposive prior expectation, there is, quote, optimality, unquote, in design terms in biological systems. Richard Dawkins is the great polemical geneticist who makes arguments that both creationists and biologists have great deals of difficulties with. So if we were playing watchmaker, if we were playing designer, and this was what we needed to do. Here's a body in whatever fashion, fly body, mouse body, worm body, whatever body, and you were to design the location of the immune system. You already have talked about what cells of the immune system do in terms of identifying and dealing with microbial incursions. So if these cells are to do this in the best possible way, where would you locate these cells? Yeah, it's a little bit like, think about the liver. The liver sits in a location that allows vascular drainage from the gut, which contains all sorts of stuff coming from the gut, to first come to the liver so that you have a filtration function. So a great deal of stuff that is coming from the liver, from the gut, is actually filtered in the liver. And for people, for example, who have liver cirrhosis and esophageal varices and bleeding, portacaval shunts used to be done. They're done less now. But what used to be done is, because the liver architecture is disrupted and therefore you have alterations of hemodynamics and therefore bleeding all over the place, liver circulation is bypassed by directly connecting gut vasculature and systemic vasculature. What that does is dump all the gut drainage, the stuff that is being absorbed from the gut, without the liver filter directly into systemic circulation, and that has its own consequences in disease terms. So this is what I mean by locational considerations. So where would you locate the immune system if the function that you are expected to design for is dealing with microbial incursions? What sort of principles for design in three-dimensional space location would one think about? So, simply to paraphrase that answer, what we would begin to ask is, what is the most likely point of entry? Is that fair? I mean, what implicit in your saying the gut is the point, you're thinking, what is the most likely point of entry? And the most likely point of entry is, of course, all those surfaces, not simply the gut, but all those surfaces where the organism abuts the external environment. So you think about that, and then you come to the next. So sure, that's where we should have as much immune function as possible. And in fact, we do. There is an enormous amount of diverse immunological combinatorial configuration in barrier, what we can generally call barrier tissues. The skin, the gut, the respiratory lining, and so on and so forth. So the next question, and that question I'm going to illustrate by a silly example. So you're walking in your ecosystem, and I recognize that this feels like an ecosystem, but this is not really our ecosystem. Our ecosystem is out there in the real world, like walking through water yesterday. So you're doing that, and you put your hand somewhere, and you get a thorn prick. Sitting like angels on the tip of the thorn were 10 bacteria. Right? So of those, because the thorn has pricked your skin, 10 bacteria, 5 have gone in. You've pulled the thorn out, 5 have been left behind. Right? So the point now is to think through what sort of configuration of what I'm calling system design would allow these 5 to be dealt with as rapidly as possible. Why as rapidly as possible? I don't know if Vinita said this yesterday, so I'm just going to reiterate. Why do we need to deal with this as rapidly as possible? So here's the issue. The immune system, if you think about all the mechanisms that Vinita talked about, that the immune system recognizes microbial targets, cells recognize microbial targets, cells engulf microbial targets, and those microbial targets are then held in acidified little vacuoles, bubbles inside cells, and hydrolytic enzymes are poured into it and they're digested. That entire cell biological mechanism is also relevant in a major non-immunological context in body design. And that is, when bodies are formed from the single cell embryo, the sperm and the egg meet, you've got one cell, and then from that one cell, X trillion cells are made. In that process, cells must proliferate. Cells must divide and grow to make from one cell this many cells. But because organs are being shaped, and I mean that literally, in three-dimensional space, they're taking particular configurations. None of those are easily possible without sculpting. In other words, without cutting away some stuff. In other words, without cells dying off. And the best example of that everybody is familiar with from school time textbooks is finger webs. Right? During development, there are webs, the web dies out, and you have digital separation. So when cells die, what is left behind? What's left behind is cellular debris. And what I mean by debris is non-aqueous phase particulate material. So that particulate material is just going to hang around unless it's cleared. And you will appreciate that the same mechanisms that you think of as being necessary for clearing bacteria, by recognizing them, by engulfing them, and by degrading them, hydrolyzing them in contained compartments, is the mechanism that you would use for dealing with cellular debris as well. Exactly the same. And in fact, there are interesting receptor overlaps between the two target populations. However, here's the difference. Supposing you let the debris hang around for a while before you deal with it. How much harm is it going to do? It will do some, because it will begin to degrade, it will make even more potentially toxic stuff, and so on and so forth. But the increase in its danger level is likely to grow at a slower rate than, to go back to the point, if you leave microbial incursions unattended. Because the great difference between the live and the dead in this context at least, is that the live will proliferate. And as microbial incursions proliferate, they become harder and harder to deal with, simply as a matter of numbers. And therefore, it's possible to begin to make the argument that immune mechanisms may actually be shaped by kinetic considerations, by needing to be quickly responsive as a more demanding feature, rather than needing to be perfect. The last one is killed. Yes, you're aiming to perfection, but you say, Acha, I have now met the target and I'm going to go away and I'm going to think through everything. I'm going to come up with the most perfect solution. I'm going to come back 2 days later and I'm going to implement it. 2 days later, the world looks completely different. In all likelihood, you're dead and there's nothing to solve. So that's what I mean when I say we're really talking about a quick response as an important feature of a plausibly successful immune response. If that's the case, then go back to my argument. 5 bacteria have been deposited under the skin, right? What are the features that will allow us to deal with them as rapidly as possible. Where do we locate the cells? What are the other considerations? What would we get the cells to do? This is how I think about modeling. What do you want? What's the perfect thing? Bacteria have been deposited, right? And right there is an immunocyte, an immune cell. That's perfect because they come in, are immediately recognized with no time lag, and whatever the immunocyte is going to do, it's going to do. Right? This is ideal. In order for this to be the highest probability event when thorns prick you anywhere, what needs to be done? You need to have an immunocyte everywhere. You ain't going to have an immunocyte everywhere because if you have an immunocyte everywhere, you don't have space for putting anything else. Correct? So how do you solve this? You need the immunocyte. You need the immunocyte to see target as rapidly as possible, but you can't put an extraordinary high density, a continuous sheet of immunocytes all over your barriers because your barriers have other functions, other cells, other requirements. So what's the trade-off? What do you do? It's the classic dilemma of how policing is designed, how beat policing is designed. So beat policing is designed by, you can't put a policeman everywhere. You can't. So what do? Policemen walk beats, right? How do you design the beat? What are the considerations that go into designing the beat? And what I mean by designing the beat is how do you design the trajectory of the walk, the speed of the walk, and the number of beats that intersect. And yet you'll appreciate that all of these features matter in being able to respond with the shortest possible practicable whatever lag time to any incursion or to anything else that policemen deal with, but in the microbial context, incursion. So what are we saying effectively? We are saying that in the tissue, in barrier tissues, we will have a certain number of immunocytes, that these immunocytes will migrate, and the number of immunocytes, their rates of migration, and their trajectories, their directions. I'm trying to simplify this as much in my own head as I can, will all be features, quantitative features, determining the efficiency of detection of microbial incursion. Is this making sense to everybody? Now, you would think that since it is self-evident to all of us that microbial incursions are likely to come like this through barrier tissues, it is self-evident to all of us that having immune responses available at the location with the shortest possible lag time is a very good thing. It's also evident to all of us that the kind of design features for immunocyte behavior that I just described to you, their numbers, their rates of migration, their directionalities of migration, and so on and so forth, would be major determinants of the success of this strategy. You imagine that one should be able to model this. and that one should be able to set, if you like, constraining values on what's possible, what's sustainable, and what's not sustainable. And then, because now we are talking about situations and properties that biologists understand reasonably well, namely, what determines migration rates? Cellular motility, the cell biology and the molecular biology of cellular motility is reasonably well understood, to the point that we can actually now begin to say, let's modulate it. And therefore, let us test whether quantitative models in which cellular motility is a determinant, whether specifically and selectively altering cellular motility alone will fulfill the quantitative predictions of a model. And surely they won't. We will go back, we will iterate, and so on and so forth. So what I'm trying to do is to suggest to you that in the simplest possible model of the immune system, which is, as I said, barrier tissues, microbial ingress, immunocytes, primarily immunocytes for those who have any biology, primarily immune cells, immunocytes, functioning as phagocytes, meaning cells that eat microbes and dispose of them. In this very, very simple-minded model, there is, I think, an extraordinarily interesting open space for trying to connect wet-work and modeling. This is even more so because the kind of genetic modifications that are unequivocally possible to achieve to change specific properties are best done for these kinds of situations in model organisms such as the worm or the fly. And in both of those, we actually do have evidence that breaching the barrier does allow patrolling immunocytes to deal with the incursions. So there are actually wet-work experimental systems quantitatively available to test these kinds of situations. So I suggest that here is a problem that we haven't actually thought about in modeling terms, and that we might want to think about it a little further. Now, that's one example. I'm going to move on from that. I'm perfectly happy to talk about details of these if anybody wants to, but if everybody is simply going to look at me, then we will drift onwards. So here's, yeah. So think about, it's a great question. What do I mean by trajectories? Now, think about the issue of cells moving. One is, of course, the rate of migration. But perhaps even more interesting is the directions they take, and because they are in tissue, they're never going to be unidirectional. So they are going to crawl in a sort of a quasi-random walk. But the walk can be shaped, and it can be shaped because cells don't swim. Cells crawl. There's a difference. So, let me give you an example. From high school, everybody's done high school biology, right? So everybody remembers single-celled organisms, right? And everybody remembers this Star Wars picture of a single-celled organism called paramecium, which is full of cilia, which can beat, and which swims directionally. Everybody also remembers our favorite single-celled organism, amoeba. So here's a question. I take a liter of tissue of medium. In fact, I can take a liter of normal saline water. And at the surface, just under the surface, precisely in the middle, I put one amoeba. Amoeba can move. Everybody knows amoeba can move. So I put that one amoeba right there under the surface of the water, right? So it's like taking a person and putting him equidistant between Chennai and Port Blair in the water, right? The person will swim directionally, little bit of a random walk, but the person will desperately swim directionally. Will the amoeba swim directionally? Why not? If I put a paramecium, cilia beating, will a paramecium swim directionally? Yes. But the amoeba won't. Why not? Because the amoeba moves by rolling over something. And what does that mean? Rolling over something means that it has to have something solid to roll over. Amoebae crawl, they don't swim. That's the difference. We're talking really about cells. You see, tissue is made up of cells and extracellular matrix, which is essentially solid stuff. And when cells move, it's not that they never float, they do. But by and large, cells of the kind that we are talking about crawl. And what changes the properties of crawling? How many people have been to supermarkets, not supermarkets, what are they called? I'm too old to remember these things, malls. How many people have been to malls where for children there is a rock climbing thing, correct? Think of that as crawling. And for small children versus teenagers showing off, the walls are different. And in how many ways are the walls different? One is that the height of the wall is different, of course. But something else about the wall is different. The handholds are much more for the little children than for the older children. In other words, the properties of the surface, the extracellular matrix over which cells crawl, will determine both the rate of migration and the choices made about directions by cells. And because of that, you can think about influencing and modulating trajectories. In other words, you can build a crawling landscape and you can alter the crawling landscape, right? Which is why, so long story short, I would argue that part of the model would be a set of criteria by which what I'm calling random walk optimalities are achievable or not. And this is biologically sensible because it's biologically feasible. Because you can make gradients, for example, with chemokines and related signaling molecules, which will attach themselves to extracellular matrix in gradients, and cells will respond to that gradient and therefore crawl in a different direction. Then they'll go from the sphere of influence or from the radius of influence of one molecule, one chemokine to another, and you can therefore shape the path that they take. You can shape the speed and you can shape the path. And in all this, we're still really asking the question of how will you model such a situation with regard to optimality of function? So what is the surrogate for optimality of function? And the surrogate is, what is the time in which every point in the interface is sampled? And how do you minimize that time with the smallest number of cells, with maximal optimization of migrational designs? So that's sort of one set of worries that immunologists have that I think haven't really been addressed in interesting ways. But let me take that forward into a parameter in that conversation that I haven't touched, and that is, I've been talking about cell migration and directions. But clearly, even though it is true that we can't put one immunocyte at every possible point in the barrier, that's not possible, we can still modulate numbers. And that brings me to a general issue that cells of the immune system, the numbers of cells available will be major modulators of the success of immune responses. Clearly, in this model, modulating the numbers, and modulating numbers again is a situation of which the biology is not too badly understood, and therefore actually doing experiments where quantitative numerical variation can be controlled of immunocyte numbers is quite possible. So, what controls immunocyte numbers? And I'm going to use a sort of set of our own data, or a description of our own data, since the details are really not germane, to make a point about immunocyte numbers that's quite different from this model. This is, I repeat, this is a very simple model. Microbes coming in, phagocytic immunocytes, looking for them as quickly as soon as possible and dealing with them, and therefore what are the quantitative parameters that need to be optimized, interlocked, and modeled and tested, and what are the evolutionary significances of such an effort. But think about it in more general terms. And in order to make that point, general terms about cell numbers and controlling cell numbers in the immune system. And in order to make that point, I'm going to tell this as a story of a PhD student research project. So, one of the things that Vinita, I, and another colleague, Anna George, run a joint academic laboratory. So, one of the advantages of running a joint academic laboratory is that the three of us almost never agree, and from the disagreements, little exploratory projects get seeded, and really it's the exploratories that are the most interesting. The dreary conventional stuff is really very boring. So, one of the exploratories that we were doing involved, for reasons that have nothing to do with the story itself, involved taking normal, healthy, young adult blood and characterizing immunocyte lineages, leukocyte lineages, subsets, subpopulations as extensively as possible in quantitative terms. We are looking for various metabolic correlates and so on and so forth. Now, every time laboratories say, oh, we took healthy young adults, right? You know whom they're talking about. PhD students. Come on. Wake up. So effectively, therefore, National Institute of Immunology, you've got hostel, you've got a bunch of PhD students. Everybody is between the ages of, whatever, 22, 28, that general range. Like, is that familiar? Okay. So, and everybody's been living there in those rooms with, oh dear, that mess food and that lab for 1 year, 2 years, 3 years, 4 years, whatever it is. Some poor, sad unfortunates even for as much as 5 or 6 years. And so we took blood samples, right? One little story from the blood sample. So there are T lymphocytes in blood, And you've talked about what T lymphocytes do to some extent. So T lymphocytes come out of the thymus. Each T lymphocyte has a different receptor, has a receptor that has a different target specificity. So long as they don't meet that target, we call them naive T lymphocytes. When some of them meet their own targets, they respond, and response means they proliferate and they differentiate. And at the end of this complex set of proliferation, differentiation, and interactions with whatever microbial targets and so on and so forth, they are now, what you are left with is a numerically expanded, functionally distinct pool of T lymphocytes. But remember, so one T lymphocyte from the naive compartment will become, say, 5, 10, 30, 100, something like that. Okay? And because they are differentiated, their cell surface marker profile is measurably different from the cell surface marker profile of the naive T lymphocyte. This is straightforward for everybody. So in CD4 T cells and CD8 T cells, you can differentiate each compartment into naive cells and let us call them memory cells. Whether the term memory is precisely applicable or not, we shall not worry about. Experienced cells, memory cells, right? So, you can now score naive and memory in CD4 and CD8. You can estimate, you can enumerate. So, you take 30, 40, 50 young men and women, and you score the proportion of CD4 T cells that are memory and the proportion of CD8 T cells that are memory. You can do exactly the same thing with B cells. You can score the proportion of B cells that are naive and the proportion that are memory. So you take all these three broad lymphocyte compartments and you score the proportion of memory cells in each compartment. So here's what happened. In this group, some people had only about 20% of their CD4 T cells as memory. Some people have as much as 80%. This is true in the CD4 compartment, this is true in the CD8 compartment, this is true in the B cell compartment. What the hell is going on? Why is there so much variation? So, after a great deal of acrimonious PI debate, PI debates are never non-acrimonious. After a great deal of acrimonious PI debate, we said, this is trivial. Why is it trivial? It's trivial because this number must be fluctuating from day to day in the same individual, and so then it doesn't mean anything. So we said, okay, we'll test this. So we tested this in a group of individuals. In other words, this particularly unfortunate group of PhD students was bled repeatedly. Correct? Because the question is, do these numbers change from day to day to day? And the answer is no, they don't change. In a given individual, over weeks and months, they stay the same. Okay? So why are they so different? So he said, somebody said, actually one of the students said, no, this is because it's very obvious. Normal memory is generated in response to infections, of course. So it stands to reason that the more infections you are exposed to, the more memory you will generate. And that is clearly the explanation. And therefore, this is pointless. So we said, oh, very good, very good, very good. So we forgot about this. And then another student went and did a little correlation. These are notions that wet bench people are really not familiar with. So they don't do them quickly. So we went away. So this young lady went and did a correlation. And what was the outcome? So she came excitedly and she said, we have a problem. So what is the problem? She said, here's the point. We are saying that this variation is explained by some people having been much more exposed to microbes than other people. So in the first place, in the past 3-4 years, we've all been living in the same hostel, working in the same lab, and eating in the same mess. But never mind, it's possible. But if that is the case, here is the problem. And the problem is that if you think about bacterial or even viral exposure, when you make antibodies to bacteria or viruses, 99+% of the times, these antibody responses that B cells will make are T cell dependent responses. They depend for being optimal in a variety of ways on a simultaneous stimulation of CD4 T cells, and B cells and T cells cross-talk to each other, and the B cell response is further magnified and refined in a variety of ways. So, if the variation in CD4 T cell memory frequency is as a result of differential microbial exposure, then B cell memory frequencies and CD4 T cell memory frequencies should be correlated. You expect that? Correct? Is everybody following this? I'm trying to simplify this without dumbing it down. It's a straightforward position. You have bacterial exposure. Bacterial exposure will generate B cell responses that are CD4 T cell dependent. Therefore, if the variation in memory frequencies is a consequence of variations in bacterial exposure or viral exposure, let us say, then CD4 memory frequencies and B cell memory frequencies should show a correlation. They don't at all. Another problem, less severe but still a problem, and that problem was CD8 T cells are thought of as being primed by viruses. None of this is absolute, and there are a lot of caveats to this, which is why I said this is a relatively less major problem, but for what it is worth. CD4 T cells are primed from locations in intracellular vesicular niches. CD8 cells are primed from locations in intracellular cytoplasmic sources. So you expect, therefore, that if at all memory frequencies should be non-correlated, CD4 memory and CD8 memory, you expect no pressing reason for them to be correlated. And damn it, they are correlated. Individuals who have high CD8 memory frequency have high CD4 memory frequency. Individuals who have high CD4 memory frequency don't necessarily have high B cell memory frequency. Nothing that you expect in simple-minded terms from a microbial load explanatory holds good. So we have a problem. So what is the source of this variation? If it's not the environment, as in frequency of microbial infections, then you're sort of forced into beginning to think that there may be a genetic component to this. In other words, that this may be a quantitative trait, genetic quantitative trait. So is it a genetic quantitative trait? So he said, how are we going to do this? So a young scientist working in the group had what I still think of as a really nice idea. He said, you know, we have this bunch of PhD students. More than half of them are from Delhi. Generally true. And all of them have siblings who are within 2-3 years, 3-4 years of age, right? All we have to do is to collect siblings. They're not twins, but they are genetically related. So then the prediction was, if we calculate memory frequencies, CD4, CD8 and B cell memory frequencies in siblings of each other, then the prediction is that if there is a genetic and or a maternal component, maternal components are not necessarily genetic components, but a sort of developmental, embryological component, then the memory frequencies of siblings will be much closer to each other than the memory frequencies of non-siblings, as a straightforward statistical expectation. And damn it, yes, memory frequencies of siblings are in fact closer to each other than memory frequencies of non-siblings. So two questions arose. One is, this is just all still correlation. Can we prove this? But the second question is, and I'll answer the first question to some extent, but then I'll ask you the second question. Because the second question is, why on earth would there be this kind of a trait polymorphism? I'm not even going to call it genetic polymorphism, but a trait polymorphism in populations. So let me answer the first question. Is this really the case? Now you can't, PhD students think that that people can and do experiment on human beings, but the fact of the matter is that you can't really do too much of it anyway. So you turn to model organisms. So remember what we are looking at. We are looking at an outbred population in which we are taking 30, 40, 50 individuals, and we are looking at memory frequencies in these individuals. Presumably the genetic diversity as a result of outbreeding in these individuals is wide enough to reveal these quantitative traits. So if that's the case, can we do this in mice? And the answer is not very well. And the reason for that is that, with a few laudable exceptions, all mice experimentally used, mouse strains experimentally used, are descended from a very narrow breeding bottleneck about 100 and something years ago, 105, 110 years ago. In the early years of the 20th century, so-called fancy mice were being bred for coat color as pets, sold as pets to kids, and then, usual. I have a white mouse, you only have a brown mouse, and therefore I'm somehow superior to you. It's like we used to trade marbles like this. I don't know what you guys traded. But so you've got a situation where most of today's mouse strains have been born from that relatively narrow bottleneck of a few mice from which coat colors have been bred, and then eventually all sorts of other traits have been bred, and so on and so forth. But it's still at its root a very narrow genetic diversity. Yet you have some. So you have a bunch of inbred mouse strains. Remember, an inbred mouse strain is, for our purposes, not for all purposes, but for our purposes, the strain is an individual, because all members of the strain, all the mice that you breed from the strain, are genetically close enough to be considered practically identical. They're not really, but still. And so the question is, how many independent inbred strains you have? Because then effectively what you have is you have that many individual mice to study for the kind of work that we're talking about. So it turns out that there are, depending on how you count them, there are anything between 25 to 50 independent inbred strains. We in the National Institute of Immunology Animal Facility have about 15, 20 of them. So we said, okay, let's look at the 15, 20. And lo and behold, these 15, 20 independent inbred mouse strains, which are housed in the same clean mouse facility, same age, same everything, show a great deal of variation in the frequency of memory in their CD4 and their CD8 compartments. For a variety of reasons, in the mouse, scoring B cell memory is not straightforward. Okay? Some mice, so if you compare pairs of strains, some show differences in their CD4 compartment memory, but not in the CD8, and some show the other way. So, a very brave student in the lab, there are no Sardars in the room. Sardars are very brave people. So, in keeping with that, a very brave student in the lab went ahead and did a relatively complicated experiment that's given results that I'm still trying to wrap my head around. So, here's what he did. You've got two strains of mice, right? The memory frequency in the CD8 compartment differs between these two. The memory frequency in the CD4 compartment does not differ. Okay? So now what he did was he mated a male from this strain with a female from this strain, and he made, in classical Darwinian terms, F1 generation mice. Okay? But his interest was not in the F1 mouse itself. What he did was he irradiated the F1 mouse so that its own natural bone marrow was killed off, and then he treated the F1 mouse with bone marrow, half of which is from one parent and half of it which is from the second parent strain. So now you have a chimeric mouse. You have a mouse in which half the T lymphocytes are genetically from one parental strain. The other half of the T lymphocytes are genetically from the other parent strain. But they have all grown up now 2 months after, you transfer the bone marrow, then the mouse becomes, the bone marrow takes and cells proliferate and everything repopulates and the mouse becomes normal. The mouse acquires whatever memory it is going to acquire because there are gut bacteria that are generating memory, this, that, and the other, right? At the end of 2 or 3 months, you now ask the question, in this chimeric mouse, what is the proportion of memory in the CD4, CD8 T cells from parent 1 and what is the proportion from parent 2? So, we are asking the same question. What we are asking is, do the two parental mouse strains show differences in CD8 memory, no differences in CD4 memory? But now, instead of asking the question in independent separate mice, we have put the bone marrow cells together and allowed all the T cells to be born and responding next to each other in the same mouse. This, therefore, completely eliminates any possibility of differential microbial exposure, this, that, and the other, because they are in the same mouse, right? And
so, won't this be because of the polymorphism in the HLA genes between the humans, like that each one have a different CD4 and a CD8 count?
so, in the first place, HLA genes don't show any differences in CD4, CD8 counts. So, they are not associated. In the second place, because of that, so I didn't go over it, because of that, the two strains that we've picked actually have identical HLA molecules. They still differ in their proportions of memory. It's a perfectly good question, but they still do differ. So, when you do this, it turns out that even when they are responding in the same mouse, cells of one parent make much more CD8 memory than cells of another parent. And what this says is two things. Number one, it says that there is actually a genetic component And number two, that the genetic component works in cell intrinsic fashion. In other words, it's nothing to do with the rest of the body, because the rest of the body is the same for CD8 T cells from the mother's strain versus CD8 T cells from the father's strain, because they're all responding in the same mouse. So the microenvironment is the same. So it's not microenvironmental differences that explain this. It's actually something intrinsic to the T cells themselves that defines this. But here's the more interesting question. We can think about, what the cellular molecular pathways are and what the genes are and this, that, and the other, and that's all okay. Here's the real problem I have. How on earth does this diversity of a quantitative trait get fixed in normal vertebrate populations? In other words, what is the advantage? So when you think about what is the advantage of having high frequency memory versus low frequency memory, we are talking about balancing selection kinds of situations, but when we still need a plausible biology to this. Are there advantages and disadvantages to having high frequency memory? Are there advantages, disadvantages to having low frequency memory? And all that frequency means is different cell numbers. That your cell number compartment size for naive T cells and memory T cells is either larger or smaller. And this is how it connects to what we were talking about earlier in terms of. Stop? Yeah, it's a complicated. I know. No, it's fine. It's fine. So the question in an evolutionary selection pressure context is, what are the advantages of having many naive lymphocytes, few memory lymphocytes? What are the disadvantages? And what are the advantages of having relatively many more memory lymphocytes? And what are the disadvantages? And we'll talk about that, because I think that it's modelable. Okay?
Okay, so since you know that it's all wet out there, we have to do this in 2 steps. So come out and stand